So having looked at the monosaccharides, having looked at the disaccharides, we need to look at the polysaccharides. The polysaccharides. The polysaccharides. So what are polysaccharides? So the polysaccharides are long polymers. So they are polymers that are made up of monosaccharides, which have been joined through the reactions of dehydration or condensation. Remember, we explained when we looked at macromolecules what dehydration reactions and hydrolysis reactions are. So these polymers are made up of or consist of monosaccharides, which are joined through or by dehydration reactions. Dehydration reactions. The polysaccharides are actually the most abundant carbohydrates that we find. Are the most abundant carbohydrates, and examples of these polysaccharides are starch. Starches, we have glycogen and cellulose. So examples include starch, glycogen, cellulose. We have also other examples that we are going to see further. So we find that the starch, as we explained, as we noted during the class on macromolecules, we said that starch is a storage polysaccharide that is found in plants. So starch is a storage polysaccharide, and it consists entirely of alpha glucose molecules that are linked in long chains. So starch consists of entirely alpha glucose molecules in long chains. Then cellulose. Cellulose is a structural polysaccharide. So starch storage polysaccharide. Cellulose is a structural polysaccharide. And it also consists of glucose molecules linked in chains. But these, however, are beta glucose molecules. So remember, starch, alpha glucose molecules, cellulose, beta glucose molecules. Starch is a storage material, cellulose is a structural material. So because starch is built from alpha glucose, we call those linkages as alpha linkages, while the linkages in cellulose are beta linkages. Beta linkages. So functions of polysaccharides. So of importance is that polysaccharides can either be for food and energy stores. So examples of these are starch and glycogen. So starch and glycogen are examples of polysaccharides which are used for energy stores and food, for food. And then polysaccharides can also be used as structural materials, structural and protective materials. So this includes cellulose and also chitin, cellulose and chitin. So this could be or act as structural and protective materials. Other functions of polysaccharides include them. They are saccharides that can lubricate skeletal joints and also able to provide adhesion between molecules. Then the other function is that these sugars, the carbohydrates, can be attached to lipids and proteins. So we can have glycolipids and glycoproteins. So these are going to act as signals. So they can act as signals. So polysaccharides can act as storage materials It can act as structural and protective materials. Examples of storage polysaccharides, we have starch and glycogen. Structural, we have cellulose and chitin. And we're going to see some other example under structural as we go further. And then we have other functions, which are lubrication of skeletal joints and also them acting as signals when they are attached to lipids and proteins. So what makes a starch and glycogen suitable to be energy storage materials? What makes starch and glycogen suitable to act as energy storage materials? So they are good energy storage materials because one, they're insoluble in water. So starch and glycogen are insoluble in water. Then two, they do not affect the osmotic potential of the cell. So they're not going to alter the osmotic potential of the cell. So henceforth, they are suitable for energy storage. And then the other property that we're going to see is that they are actually highly branched. They are highly branched and are able to fold into compact shapes within a very limited space. In a very limited space. And then we also note that these polysaccharides are able to be converted to disaccharides and also to monosaccharides through the process of hydrolysis. So starch and glycogen can easily be converted back to disaccharide molecules and also to their monomers, the monosaccharides. Then we have cellulose and chitin, which are actually structural materials. So what makes them suitable for being structural materials are the properties of them being insoluble in water and also them being made of long threads or fibers. So they're made of long fibers or threads. And then they also have a high tensile strength. So these are properties. So they are insoluble in water. They have long threads or fibers, and also they have high tensile strength. So these are the properties that make starch and glycogen to be energy storage molecules, and cellulose and chitin being suitable structural and protective elements. So let's look at starch. Let's look at the polysaccharide starch. So we find that starch is actually the typical form of carbohydrate that is used for energy storage, as we have mentioned. And this energy storage is in plants. And we mentioned in the preamble that it consists of alpha glucose subunits. So it has alpha glucose molecules, which are joined by a 1,4 or 1 to 2 linkages. So it's one, rather they're joined by a 1 to 4 linkages. So alpha glucose molecules joined by 1 to 4 linkages. So this means that carbon 1 of one glucose molecule is linked to carbon 4 of the next glucose in the chain. So no wonder it's called 1,4 or 1 to 4 linkage. So carbon 1 of one glucose molecule linked to carbon 4 of the next molecule in the chain. So what happens for starch to be formed? Remember, it's a polymer. So there are more than 10 monomer units or alpha glucose monomer units in starch. Now for that chain to be formed, firstly, these the alpha glucose monomers will first join to form maltose, the disaccharide maltose. So we are going to have maltose units. And then these maltose units will then be joined to form the starch. The maltose units will then be joined to form starch. So this means that when there's complete hydrolysis of starch, complete hydrolysis of starch will lead to the formation of alpha glucose. And there's complete hydrolysis of starch, what will be formed is alpha glucose molecules, since the glucose molecules are the monomers. So when you look at starch, starch occurs in two forms. So starch occurs in two forms. So we have amylose and amylopectin. So amylose and amylopectin, these are the two forms of starch. So when you look at amylose, this form of starch is unbranched. So take note that amylose is unbranched. And then, as I mentioned, it's a starch, so it would compose hundreds of alpha glucose molecules that are linked together in long and unbranched chains. So long and unbranched chains. And then it contains only alpha 1,4 glycosidic linkages. Remember the linkages or the links that we find in carbohydrates, glycosidic linkages. So amylose will contain alpha 1,4 glycosidic linkages. So this occurs between carbon 1 of one glucose molecule and carbon 4 of another, which leads to the formation of this alpha 1,4 glycosidic linkage. Then amylose is insoluble in water because of the long chains of amylose that tend to coil up in water. So those amylose chains are able to coil up in water. Hence, this makes amylose to be insoluble in water. When we suspend amylose in water, it's going to give us a blue-black color with iodine. It's going to give us a blue, a black color with iodine, because because it's going to form a complex, or rather, the iodide complex is going to slip and go into this alpha helix coil, hence giving that blue-black color. So note that starch is about, it contains 20 to 30% amylose. So starch is about 20% to 30% amylose. So now for the synthesis of amylose, remember we said that starch will be formed by maltose. So glucose, alpha glucose will first form maltose.
It was unused. It was unused. So I have to fix.
Okay, so we said that starch will first form maltose, and then these maltose molecules will then be linked through the process of dehydration to form amylose. So remember, amylose is a strand chain. These molecules are connected by alpha 1,4 glycosidic chains or linkages. So first maltose, and then those maltose molecules join to form amylose. So when there's hydrolysis of amylose, we're going to have maltose molecules, and then with further or with complete hydrolysis, we're going to have alpha glucose molecules. Alpha glucose molecules. Then we have the other form of starch is what is called amylopectin. So this is more common, and it is branched. So it contains a branched chains. So the branching that we find in amylopectin will occur when glycosidic linkages form between carbon 1 of a glucose monomer on one strand with the carbon 6 of a glucose monomer on another strand. So this means that we are going to have 1,6 glycosidic linkages together with 1,4 glycosidic linkage in amylopectin. Unlike amylose, where we only have 1,4 glycosidic linkage. So now when we look at amylopectin, if we suspend it in water, it's going to give us a red-violet color because we find that there's little interaction that occurs between this iodide complex and the amylopectin. Unlike in amylose, where we find that this iodide complex is able to slip through the coil of the amylose. So amylopectin is branched. Of note is that the branches that occur in amylopectin will happen 1 out of every 30 monomers are going to have a branch. 1 out of every 30 monomers are going to have a branch. So this one will become especially important when we start looking at glycogen, when we compare amylopectin and glycogen branches. So remember, amylopectin is the most common form of starch. So starch will contain about 70 to 80% of starch. It will contain 70 to 80% of amylopectin. So 70 to 80% of starch is amylopectin. Amylopectin. So this is the breakdown or the dehydration, the synthesis through dehydration reaction and the breakdown through hydrolysis. So you have amylose chains. These amylose chains, through the process of dehydration, we have the formation of this 1,6 alpha 1,6 glycosidic bond. So this is forming this chain. So we have chain 1 and chain 2. These chains, remember, carbon 1 of a glucose monomer on one strand and carbon 6 of glucose monomer on another strand link to form this alpha 1,6 glycosidic bond. Okay, so we find that when we look at starch, we find that plant cells are able to store starch mainly as granules in organelles that are called amyloplasts. So amyloplasts are able to store starch. So when the energy is needed by the plant for the cell to function, the plant is able to hydrolyze the starch. So when that hydrolysis happens, this is going to yield the glucose subunits. So I find that organisms, including humans and animals, actually they are able to break this alpha 1,4 glycosidic bond or linkages. Furthermore, when you look at starch, it is insoluble in cold water. It is insoluble in cold water because of its being a long molecule with many glycosidic bonds that form between the glucose monomers, and also because it is tightly packed. It is a tightly packed molecule. However, we find that starch can become soluble in water when it is heated. So what happens is that those granules are able to swell and burst So that a semi-crystalline structure of starch is lost, and henceforth those are smaller amylose molecules are able to start leaching out of the granule. So they're going to form a network that holds water, and henceforth are able even to increase the viscosity of water. So starch can become soluble in water when it is heated. So this is simply a process that they call, or the process which is called starch gelatinization. So through starch gelatinization, starch is able to dissolve in hot water. So remember, we said that starch is a major energy storage in plants. So we find that it is actually common in human staple food. So for example, in rice, we'll find it in rice. We're going to find it in wheat, in maize, in cassava, and also in potatoes. And then when we look at the industrial application of starch, we find that it is used as a thickening agent or a stabilizer or a prebiotic. So starch is quite important. Then we look at glycogen. It is a comparable molecule to starch in animals. So it means this is a storage material in animal, a storage polysaccharide in animals. So it is an insoluble polysaccharide that contains branched amylose chains. So just like in amylopectin, glycogen is also a branched molecule. However, glycogen has much longer chain length and more branches than the starch. So glycogen, the chain for glycogen is much longer and the branches are more, so they come frequently. Remember I said that for in amylopectin is 1 per 30 monomers, then there's a branch. But we find that in glycogen, it's 1 out of every 10 glucose subunits, there's going to be a branch. So glycogen is also a polymer of alpha glucose, and near identical to amylopectin, which is a branched form of starch. So the alpha 1,6 glycosidic linkage occurs 1 out of every 10 glucose subunits in glycogen. So this means that glycogen will be more branched, or rather the branches will be shorter and more frequent. So they will be more frequent since it's occurring every after 10 glucose subunits. You have an alpha 1,6 glycosidic linkage. And then it is insoluble because of the numerous alpha 1,4 and alpha 1,6 bonds, and also because it has a compact structure. So when you look at glycogen, it's going to occur in the form of granules that are found in the cytoplasm. However, in human beings and also in other vertebrates, we find that it is mainly stored in liver and muscle cells. So when glycogen is needed or glucose is needed, glycogen is able to be hydrolyzed to give a glucose that can be used during the process of cellular respiration. So glycogen is able to be hydrolyzed. So this is a structure of glycogen, simply showing glycogen structure. So we have the 1,6 alpha 1,6 linkage there between carbon 1 of a chain and carbon 6 of another chain, carbon 1 and carbon 6. Let's look at cellulose. So when you look at cellulose, cellulose is a structural carbohydrate, as we already mentioned. It is a structural and protective carbohydrate. So it is very abundant in plant cells. It is very abundant in plant cells, which act as a supporting and protective material. So cellulose is a structural polysaccharides that is found in plant cell walls. Plant cell walls. So cellulose is a polymer of beta glucose monomers. So it is a polymer of beta glucose monomers, which will be joined or which are joined by beta 1,4 glycosidic linkages. Now, what happens is that linkage, the geometry, we find that each glucose monomer in the chain is flipped in relation to the adjacent monomer. So each residue will be turned about 180 degrees relative to its preceding residue. So this orientation is important. This orientation is important because it implies or it makes this cellulose molecule to be a linear molecule rather than a helix or a helical molecule that we see in starch. So cellulose is a linear molecule. And then this also permits multiple hydrogen bonds that form between adjacent parallel strands of cellulose. So it leads to the molecule forming hydrogen bonds. So as a result, we find that cellulose forms long parallel strands joined by the hydrogen bonds. And the linked cellulose fibers are very strong and are able to give the cell its structural support. So this is very important for the function of cellulose, the hydrogen bonds. So those are beta glucose subunits. Remember, are joined in a way that will allow extensive hydrogen bonding to exist between cellulose molecules. And henceforth, they aggregate in bundles of fibers. So they have to aggregate in bundles of fibers. So those are neighboring cellulose molecules lie close together in layers and form rigid structures that are called microfibrils. So these microfibrils are able to aggregate to form what are called macrofibrils. So the hydrogen bonding, it's very important, the function of cellulose. It is chemically similar to amylose. However, we find that the starch hydrolyzing enzymes, which are found in most organisms, are unable to break between two beta glucose units. So these bonds cannot be hydrolyzed by enzymes that hydrolyze the alpha linkages that are found in starch, because those enzymes are specific and they can only recognize the alpha linkages. So now what does that mean? Because of that, and that because cellulose cannot be easily broken by most of the creatures, it works well to be a biological structural molecules. However, we have some organisms, microorganisms that are able to digest cellulose to give glucose. So we have bacteria, we have protists, which can be found in the digestive systems of cows and sheep. So they occur in these animals as symbiotic bacteria. So these enable these animals to be able to obtain nutrients from cellulose. So for example, other examples are termites, which also contain microorganisms that can digest cellulose. So these organisms are able to provide the necessary enzymes for cleaving these beta 1,4 linkages. And henceforth, they are able to access these rich sources of energy. So we have these animals that are able to digest cellulose. So for example, cows, sheep, have termites as well. They're able to digest these. How about in human beings or animals that may lack enzyme to digest cellulose? It means that these animals or these creatures cannot use them for nutrition. However, these are very important for the functioning of the digestive system. So the cellulose is fibrous, and henceforth it provides bulk to help keep our digestive tract functioning properly. So cellulose acts as dietary fiber. It acts as a dietary fiber. In the industries, we find that cellulose is being used to produce paper and also cotton threads. And also recent researches are aimed at converting cellulose into biofuels. So cellulose is an important polysaccharide. It is an important polysaccharide. So this is a molecule of cellulose. It is a molecule of cellulose. So remember I said that the adjacent molecules, glucose monomers, are flipped 180 degrees. So remember this is carbon 6. This is carbon 6. And then the next monomer is here, the glucose monomer. The carbon 6 is here. So this monomer, glucose monomer, has been flipped about 180 degrees. 180 degrees. So this is the cellulose molecule. Cellulose molecule. Then we have chitin. We have chitin, which is a structural material. So usually this one is found in arthropods and fungi. So arthropods, fungi, it's found in insects, in crustaceans, chitin. So it's a polymer of N-acetylglucosamine, which is a substituted version of glucose. Chitin is similar to cellulose, but it has the hydroxyl groups of carbon 2 of each glucose unit being replaced with this acetamide group, functional group, acetamide functional group. So chitin has an acetamide functional group or acetamide group at carbon 2. So carbon 2 is replaced with an acetamide functional group. So when these are cross-linked by proteins, this leads to formation of a tough resistant surface material, which serves as the hard exoskeleton that we find in insects and crustaceans. So it is a very important component of these external skeletons of these creatures. So those N-acetylglucosamine subunits will be joined by glycosidic bonds, which are beta 1,4 glycosidic linkages. Remember I said that this is similar to cellulose. So it forms a very tough structure because of the geometry of these bonds, which results in every other residue being flipped in orientation. So just like in cellulose as well, they are flipped in orientation. And these molecules are also able to interact through multiple hydrogen bonds, between adjacent strands, just like in cellulose. So this leads to chitin being responsible for provision of stiffness and also protection. So this tough sheet will provide stiffness and also protection. So we have in other organisms, where there's a further addition of calcium carbonate, which further makes this chitinous structure to be more hard. So it hardens, provides further hardening, the addition of calcium carbonate. So this is a chitin molecule. So remember, just like in cellulose, we have the flipped, it's flipped. The adjacent molecules are flipped. Then we have this functional group that has been added, or that has replaced the hydroxyl group. Okay. So we can log out and log in so that we can just finish up. We're just remaining